Hey CW Bio, Mr. Kennedy here, and this is part 2 of your Chapter 9 lecture notes on cellular respiration. Today we're going to get into the ins and outs of how energy is extracted from your food through, well, a lot of biochemistry. If we just try to sum it up in a very simple sentence, respiration or cellular respiration is a step-by-step process to slowly break down glucose to utilize as much energy as possible from it, well, safely. And no matter what we do, well, really this whole process is only about 40% efficient. That means that 60% of the energy available in glucose is actually lost as heat. And while that sounds terrible, well, it's actually important because it keeps your body 98.6 degrees all the time. So yay. And if you were like wondering, why don't we try harder, do a better job? Well, I mean, hey, we're doing okay at 40%. If I told you you only had to get 4 questions out of 10 right on your test to get an A, like, wouldn't you just like answer 4 questions and be like, yay, I got an A? That's kind of like what your cells are doing. They get an A for 4 out of 10. Okay. All right. Let's look at how this process works. Anyways, so cellular respiration can be broken down into pieces. First piece I want to share with you is called glycolysis. Glycolysis literally means glucose cutting. So we're going to take glucose, that molecule we talked about in the first part of this video series, and we're going to split it in half. When we do that, energy is released, and believe me, a lot of it. So we want to be very, very careful with how we release it, so we literally don't vaporize our cells. Glycolysis basically occurs in the cytoplasm of your cells. It doesn't require any oxygen, and ultimately it's the starting point of cellular respiration, breaking glucose into 2 pyruvates, 4 ATP, and 2 NADH2s. All right, more to come on this in a minute. If no oxygen is present in your cells, and I know what you're thinking, well, I'm breathing, so how is that possible? Well, physiologically speaking, I got to tell you, it's impossible to supply every cell in your body with all the oxygen that it needs in order to do aerobic respiration, the form of respiration that involves oxygen, all the time. So there will be a time, like when you're exercising and you can't get the oxygen in fast enough, that your cells switch over to anaerobic respiration, which is what this is, aka fermentation. Fermentation occurs if no oxygen is present, and it comes in 2 forms, alcoholic and lactic acid. Alcoholic fermentation occurs only in plants like yeast, and lactic acid is what we do, like when you exercise and then you get sore muscles. Lactic acid. Yeah, not fun the next day. So, aerobic respiration. Well, this one only occurs in the presence of oxygen, and that's like the whole process of the Krebs cycle, from start to finish, beginning to end. And yeah, it's pretty dang cool. Okay, so at the end of the day, like, that's the part that we need to make sure that we understand, so we can like have an idea where all the energy in our food is coming from. So let me walk you through it. First thing we got to talk about are the steps of glycolysis. Remember, I told you about glycolysis a little while ago. Glycolysis is a stepwise process, basically to split glucose into 2. Remember, no oxygen is required, and it occurs in the cytoplasm. So let me just walk you through the pieces and see how it goes. Glucose is a stable molecule. It really doesn't want to react, so it actually takes some effort to get this molecule to split. So to cut glucose in half, we have to invest a little bit of our own energy to start this process, and that means ATP. So it's kind of like the idea of you can't make money unless you already have some money. You can't make ATP here unless you already have some ATP so that you can activate glucose. Okay, so provided you have a little bit of ATP, you're going to activate glucose by attaching phosphates from your ATP to the glucose. That's going to change its name a little bit, as you'll see on the coming slides. So glucose will be activated and its shape will be changed. Glucose will be isomerized. Isomerization is a fancy chemistry term for basically taking the atoms of an atom and reorganizing them. So we're going to take glucose from, like this nice little, 6-sided, almost circular shape, and we're going to rearrange the atoms so it almost looks like a house. it'll be isomerized into fructose. And then we're going to do a little bit more work with ATP and throw in another phosphate, okay? So at this point, our glucose, well, it's been kind of smushed and manipulated around, and some phosphates have been thrown onto it, so we can't call it like glucose anymore. Its new name, after we've done all this work, is fructose 1,6-diphosphate. Now, that molecule is super easy for us to like cut right in half, okay? And that's why we do it. Like, it's got 3 carbons over here and 3 carbons over here. It's a nice, easy split. So that's the whole point. We use the phosphates that we've tacked onto this fructose to like rip it in half, right? And as we rip it in half, like that's where we start to phosphorylate or add phosphates back to ADP and harness some of the energy from that split. We're also going to produce some high energy electrons from that split, and those high energy electrons are going to be transferred to an electron carrier called NAD. NAD stands for nicotinamide adenine dinucleotide. You don't need to know that for your test. You just need to know NAD, okay? And when it picks up those electrons, it becomes NADH2. Pretty cool, okay? Now, once it's split in half, again, it's not glucose, and it's not fructose 1,6-diphosphate. Split in half, we call it pyruvate, right? So pyruvate is what's left over after we like split our glucose completely in half. Now, in the process of making this pyruvate, you might have noticed, right? It did say on that previous slide, we're gonna phosphorylate 2 ATP and do that whole NAD thing. Well, the last thing we're gonna do is we're gonna phosphorylate 2 more ADP to make 2 more ATP. So if you're like a math person, you're like, okay, well, that's 1, that's 2, that's 3, that's 4. So we made 4 ATP in this process. 4. Yay. Okay. Problem is, it cost us 2 to start the process. So we've really only gained 2 ATP. We made 4, but we gained 2. That's a whole like 14 kilocalories of energy. Glucose has 686 total to offer. You released 14. That's like taking a final in biology and only getting 14 out of 686 on your test. Not good. And no, you can't live off that. So there has to be a better way, right? So here's your end products of glycolysis: 4 ATP, right? Net, because like 2 are used by the body, and then 2 NADH2s and 2 pyruvates. Remember what I said, if there's no oxygen present, well, then you're going to go off into fermentation. This could kind of keep you going for a little while, but like, yeah, this is kind of like taking a Prius that's supposed to get 50 miles to the gallon and like running it on its rims with no tires. Like, your fuel economy is going to go, and yeah. So you're going to burn up whatever gas and battery charge and whatever you got, and you're not going to go very far. Okay, so you can't live off of fermentation. That's why we get tired when we exercise, because you're not getting oxygen to your muscles, and you're not doing aerobic respiration, and you're not getting energy from your food, so you got to stop. So, as I said a minute ago, there must be a better way. Here's that better way. Aerobic respiration is that better way, right? Let's skip right to it, okay? Aerobic respiration. Well, this is like what's going on in your mitochondria, okay? Glycolysis occurs like out in the cytoplasm of your cell. You don't need anything special. Like, there's no oxygen like present, none of that. Like, aerobic respiration, like, this is like awesome. This is where all your energy comes from your food. It occurs inside our mitochondria, inside of our cells, but again, only if oxygen is present. Now, probably the most important part of this aerobic respiration is this critter called the Krebs cycle. It was discovered by a man named Hans A. Krebs, so there you go. It's also known as the citric acid cycle, and basically I won't bore you with all the different steps of the biochemistry and all that. You're just going to have to accept, like, you know, my word for this, but what you need to know is that this is a series of reactions that's designed to produce electron carriers to, like, use in the electron transport chain so we can get all our ATP. So in a fancy, like, kind of technical term, we're going to oxidize our fuel molecules, we're going to reduce our electron carriers so they're energized, and we're going to take those to the electron transport chain. Fantastic. Now, this is what it makes. 4 NADH2s for every acetyl-CoA that enters it. Wait. Mr. Kennedy, where'd that acetyl-CoA come from? Well, I'd be remiss if I didn't tell you, so I'm going to tell you. When your pyruvic acid goes to like climb inside your mitochondria, it's too big. Like, it has to be converted into acetyl-CoA, and to do that, we chop a carbon off of it, so it goes from 3 carbons to 2 carbons, and that carbon you chop off is, that's your CO2 that you breathe out, right? So if you ever wondered why, like, you exhale CO2, now you know, okay? So that acetyl-CoA, you know, there's 2 of them for every glucose that goes into the mitochondria. So you get 4 NADH2s per acetyl-CoA, so that basically means you're going to make 8, you know, it goes around twice. And 1 FADH2 per acetyl-CoA, so you're going to make 2, it goes around twice. And then 1 ATP per acetyl-CoA, so when the whole thing's done, you're going to make 2 because it goes around twice, okay? All right, so there you go. So the ATP can go off and do its business. The NADH2 and FADH2, we got to talk about those guys. So they're going to go off to this, the electron transport chain. The electron transport chain is, well, again, another series of reactions. This time, what we're trying to do is build an osmotic gradient so that we can pump protons like from one side of the mitochondria's membrane to another, right? You could think of it like a hydroelectric power project, a dam. Okay, so basically if I have a lot of water on this side of the dam and very little over here, right, gravity takes over and I can let the water go through the dam and make electricity. So yeah, that's what we're trying to do, but at a chemical level inside of the mitochondria. So we're trying to get lots of protons on one side of the membrane, very little on the other. We let the protons go through, and that's the power that we're going to use to take a phosphate and stick it on to ADP, right? So that's where the majority of our energy is made, right? In the electron transport chain. Super fancy. And no, I'm not going to test you on here, name that molecule as it goes through the chain. Don't have to worry about it, but you do have to know. Protons are pumped from one side of the membrane to the other. It makes a concentration gradient, and that concentration gradient is used to power sticking a phosphate on an ADP so that we have ATP. Okay, where does it occur? It occurs in the inner membrane of the mitochondria, the cristae, and we'll worry about that endosymbiosis thing later. How many are produced? Well, that's unusual. I hear a bell. How many are produced? Well, the electron transport chain produces 34 ATP from our 2 FADH2 and our 10 NADH2s. And wow, if you do a little bit of math, multiply that by like 7.3, we're talking about almost 300 kilocalories from the 686 that were originally available from glucose. So that's a huge step in the right direction in terms of extracting energy from our food. It's not perfect. It's not the best. It's not like we got all the energy that we could, but it's enough for us to survive on, right? So that's fantastic. So the energy yield, remember, is each one of those ATP can yield 7.3 kilocalories of energy. So you're going to multiply that times 34 just for the electron transport chain. Then you got to start adding in, well, what did I make in the Krebs cycle? And what did I make in glycolysis, right? So that's where we start to maximize like the energy extraction and get to that 40%. Ultimately, you can make like or keep 38 ATP per molecule of glucose. You really made like, almost you made 40, but like, it cost you a couple to start, right? So you got 2 from glycolysis, you got 2 from Krebs, and you got 34, from electron transport. So that, ladies and gentlemen, is where we get our 40% efficiency. Okay. Now, hopefully, as you watch this video, You know, watch it forwards, backwards, watch it 10 times. Like, write this stuff down. Try to teach it to your friends and your family. Like, you're going to have to spend time with it and let it soak in. It's not going to be easy to remember if you don't. So I'm Mr. Kennedy. I hope you've enjoyed this like real quick coverage of cellular respiration. I'll see you next time.